Yeah. Alright, okay. Pay attention, class. Enough. Everyone pay attention. So, yeah, what is the subject? Code? 253. Basic hematological diseases. Practical batch. Today's date 28/02/2024. Alright. I will tell some interesting things, then we will go to the academics. I don't know from where I need to start. So there is a very poor family, okay. So from a very poor family, okay, they have 8 children. This poor family has 8 children, okay. They are farmers. And they have 8 children, and this is in the 1901, 100 years ago. Class, quiet, pay attention. Listen, this is 100 years ago story. Listen, everyone. Yeah, 1901. That year is 1901, and they have 8 children. And one of their second children. This story is about the second child. Okay, I will tell about his story. Really interesting. So, he started talking. Class, quiet. This child started talking by the age of 2. He started talking, okay. He can able to express his things at the age of 2. My son says, okay. He still don't know, only Papa and Mommy he knows. Anyway, sick and cocoa melon. That's it. Only these 3 words. Anyway, so what happened? By the age of 2, he started talking. By the age of 7, he started, solving mathematical problems. He started solving maths by the age of 7. Okay. By the age of 11, he graduated from 10th class. By the age of 11, he graduated from 10th class. Keep in mind, at this time, studies is not a, it's a big deal. No one used to study at this time. They used to go for working in the farms, right? But this kid showed a brilliance in academics, and something really interesting happened. He started to understand the concept of light. What is light? Light is one of the mystery in science actually. Okay? Newton thought light is, light. What Newton did is he kept a spectrum, prism, and he got a spectrum that gives you colors. And Newton said light carrying some energy. And parallelly there is this Einstein. Einstein said light carrying energy. Whereas other scientists thought light is in the form of waves. Some said light is like an energy, whereas others said light has some particles, photons. Okay, we don't know exactly. Light behaves in both the way. Light behaves like an energy, and that means light will behave like a wave, and also light will behave like a particles. Okay, the reflection property looks like a particle nature. Light bounced off. Reflection looks like a particle nature, whereas a wave means light will pass through a glass. It looks like a wave. Anyway, these are some interesting things in science. Then, our kid, by the age of 13, he graduated from BSc. Okay, he graduated from bachelor's by the age of 13. Okay, very interestingly, this is the first child to be, graduated from Madras University. That is big university in India at that time, okay. By the age of 14, just 1 year gap. You know what happened? In 13th year, when he is doing his BSc, he published a paper in Royal Society Journal. He published a paper where Einstein publishing papers. At the age of 13, this kid publishing papers where Albert Einstein publishing. And Albert Einstein is 32 years old at that time. Okay? So very, very smartest kid. Very smart. By the age of 14, he got his master's degree. By the age of 17, he became an assistant professor with PhD. Doctor. By the age of 17. Okay? Just imagine. He got a very good support from his teachers. Teachers understood his brilliance and they sent him to the England. Okay. Now, in the England, what he did? He started to observe some interesting phenomena about light where he studied at the age of 7. He understood that the scattered light, whatever light that is scattered, that scattered light still having some energy, and how to count that energy, how to evaluate the energy of a scattered light. Is there any applications of scattered light? Okay. By understanding the mathematical principles of the light scattering, he collaborated with one scientist called Rayleigh plus this scientist. He has mathematically derived why the sky is blue. Today we came to know why sky is blue, because there is a pattern of scattering of light. That's why light is the sky is blue, and why sunset is in orange. Sunset is in orange and sky blue. Sky is in blue. Was derived by this scientist. That is a very small scale. What to say? That is a small experiment for this scientist. But he has a other groundbreaking thing, that is this scattering phenomenon. I don't know whether you can appreciate this scattering phenomenon or not. The applications of this scattering is, since we understood light has some energy, scattered light has energy, can we use this energy to develop an images? Can we make scattered images of substances? Can we able to see the invisibles with a scattered light? The invisible means the molecules. Let me say I have a tissue and I have some antigens. Antigens are molecules. Can I use a scattered light to understand the molecular structures? Can I able to use scattered light to understand DNA structure, ribosome structure, mitochondrial structure, subcellular internal structures? Is it possible? So yeah, it took next 40 years to have the practical applications of scattered light. Okay. On one day in 1928, I guess, okay, he's from India, keep in mind. Einstein took almost 40 years to get Nobel Prize. It took almost 40 years for the Einstein to get Nobel Prize. Nobel Prize is the biggest achievement to a scientist, right? Nobel Prize, it's the biggest scientific award. And he is the first Indian to grab Nobel Prize from India, okay, in the field of physics on February 28, 1928. And this February 28 is celebrated as National Science Day in India. And you know who is this scientist? He is Dr. C.V. Raman. And that's why we are celebrating National Science Day in India in contributions of C.V. Raman's Nobel Prize discovery. That is C.V. Raman effect. So the entire world came to know why sky is blue because of India and because of C.V. Raman. And the field of medicine has been drastically changed. They discovered DNA because of C.V. Raman. Rosalind Franklin started understanding the scattered X-ray light because of C.V. Raman's mathematical principles. The entire science and medical field is attributed to a single person. A single person, at the age of 7, his curiosity on light. By the age of us, he became a Nobel laureate. By the age of 30, he is a Nobel Prize winner. So that is the story of C.V. Raman, and that's why we are celebrating National Science Day. Isn't it interesting? Yeah, that's why we are celebrating. And most of the times, we are not, I haven't seen anywhere the Nobel discovery of C.V. Raman. No, have you seen C.V. Raman's photo anywhere? No. But we are celebrating this day for C.V. Raman. Please keep in mind, okay? Yeah, very big guy. So coming back to our medical technology today. Today's topic will be detection of hemoglobin S. What is HbS?
Sickle cell anemia.
Sickle cell anemia. So what is sickle cell anemia? Yeah, it is a genetic disorder. Very good. What happens in this?
Shape of
abnormal shape. Very good. Yeah. Listen. So sickle cell anemia. Sickle cell anemia. So in sickle cell anemia, the shape. What is the normal shape of the disease?
7.5.
Biconcave, very good. Biconcave shape. But in case of sickle cell disease, the shape of the RBC will be sickle shape.
C shape.
Okay, if you remember the CPI participle, that sickle, okay, the one which we will cut grass, that sickle shape, okay. Anyway, this is sickle shape. Listen, this sickle shape happen, biologically speaking. Hemoglobin has two types of peptides. Can anyone say? Alpha peptides and beta peptides. Two alpha peptides and two beta peptides. There is a problem with the beta peptide of the hemoglobin. A problem with beta peptide. A problem with beta peptide resulted in formation of sickle shaped red blood cell. Okay, in the absence of oxygen. So hemoglobin has two types of chains, alpha chains and beta chains. There is a problem with the beta chain of the hemoglobin.
In present or in absence of oxygen?
Now listen, beta chain has an issue. This beta chain prepared the hemoglobin. Usually this problem won't be seen in oxygenated condition. In oxygenated condition, the red blood cell looks like a biconcave. But once the patient got a excessive carbon dioxide or decreased oxygen, the shape of the red blood cell will change to sickle shape. So what are some normal conditions where listen, under normal ventilation, sickle cell patients can live happily because they have biconcave shaped red blood cells only. But once they got a less oxygen environment, they will have an issues. Less oxygen environment such as high altitudes. If you climb to any mountains, there is a chance of getting sickle cell complications. Now there is one complication called a sickle cell crisis. Sickle cell crisis. What is this sickle cell crisis? I will just give you some demonstration or try to understand an analogy. You have eyes, right? In the eyes there are some very small blood vessels. You just imagine these blood vessels blocked by sickle shaped RBCs and you started bleeding from the eyes and you will become blind. Okay, so this is one crisis. This is ocular bleeding, okay, because of sickle cell anemia. The blood vessels in the eyes will be blocked and you will eventually become blind. This is one condition. Second crisis in sickle cell crisis is the alveoli. The blood vessels in the lungs will be blocked with blood. The blood will start clotting in the lungs, leading to respiratory issues. Already they have issue with anemia and they are getting respiratory issue. They will die because of hypoxic shock. Okay, so respiratory collapse can happen due to sickle cells. Okay, these sickle cells will block the blood vessels in the lungs, leading to respiratory issues. Okay, so yeah, these are some things about sickle cell anemia.
Sorry, sir. The first point.
The blood vessels in the eyes will be blocked, leading to blindness. Yeah, okay. Eyes have smallest capillaries. In the capillaries, blood vessels, the sickle-shaped cells will block the arteries. The sickle-shaped RBCs will block the capillaries, leading to ischemia to the eye. That means the blood cannot flow to the eye and they will become blind. Okay? Yeah. Next one. So when I discuss about the genetics, again, this is a autosomal recessive disorder. This is an autosomal recessive disorder. That means both male and females will be get affected. Either we will be a carriers or two recessive, then they will get affected. Right? So that is all about a sickle cell. And some interesting things are there. Let us see the geographical distribution of sickle cell anemia. Sickle cell anemia is very common among African continent. African continent. Why? Why sickle cell anemia is common among African continent? The reason is in Africa there are many mosquitoes. Mosquitoes are carrying a deadly parasite called Plasmodium requires a biconcave red blood cell for its life cycle. Plasmodium, Plasmodium parasites, sporozoites, right? Menozoites. Do you have parasitology in this semester? Do you completed malaria? Not at fine. So malarial parasite will go into the red blood cell and it will cause malaria infection. Okay. Now listen, some interesting phenomenon. If your cell is a sickle cell, if you have a sickle cell disease, you won't get a malaria. Malaria parasite cannot enter inside a sickle shaped RBC. So Africans, I can say like this, the evolution favored like this in Africa. To protect the population must have sickle cell anemia. If not, they will die because of malaria. So the evolution, the gene pool in the Africa has a sickle cell trait. Most of the Africans are prone to sickle cell anemia. The reason is to get protected from malaria. Okay, there is a evolutionary selection has been happened in Africa. So the rule in Africa is: do you want to die with malaria or you want to be an anemic? You want to be a lazy person or you want to die? Be a lazy person. Listen, there will be some anemic problems only. They will be somewhat lethargic, weak, but they won't die. Sickle cell anemia won't kill the patient unless it is entering into the late stage, crisis. When crisis happens, if these people climbing into any mountains, if these people go into less oxygenated conditions, if these people diving into the oceans, where they are holding their breath, but under a healthy lifestyle, these patients will be good. They won't have any issues under healthy lifestyle. Yes, Mrs. How sickle cell prevent malaria? I will tell very simple. You can easily enter into this room. Can you able to enter into this cupboard? Easily you cannot, right? So that cupboard is like a sickle cell. Malaria parasite cannot enter into a cupboard, but it can enter into a room. Simple. It has a entry pathway. The door is opened in the biconcave shape per cell, but door will be closed in sickle shape. So it cannot enter inside. That's it. I said in a very layman terms, but there are some receptors, some pathways are there, okay? But understood, right? Parasite requires a biconcave RBC, that's it. That depends, maybe. Yeah. Anyway, very good. Yes. Then, any more points I need to discuss? Yes. Now coming to the test. First I will tell the simplest test procedure. Then we will discuss the depth test procedure. Okay. So the class of Cornwall not having PPE. Then Abhiram took my permission. And Palak not having mask. Keep your PPE properly, everyone. I'm getting irritation. See now he is returned. That's it. Keep buttons to the lab coats, everyone. Very bad students. Okay. Pay attention. Very simple. If I suspect a patient has a sickle cell anemia, first I will collect his blood. Now, when I perform CBC or PBS, when I perform the peripheral blood smear, can I catch a sickle cell patient? Yes. I cannot. His cells looks normal. In PBS. In CBC, he might have a reduced RBC count, might be, but in PBS, in peripheral blood smear, we cannot see any sickle cells. You understood why? Because at room temperature at sea level, oxygen is more. Patient has a normal oxygen condition. So how we are expecting a sickle cell? That means sickle cell is an asymptomatic disease. Even though the patient is a sickle cell patient, under oxygen conditions, we can't catch them. They look asymptomatic. You got my point? Sickle cell requires less oxygen environment. Then only the cell will be, RBC will be converted to sickle shape. So how to perform this test? PBS is not an ideal approach to identify sickle cell disease. Then what should we do? Yeah. Now what we will do is, now I will add his blood in a test tube. In this test tube, I kept a redox solution. Redox solutions means what happens? Reducing solutions. They will remove oxygen from the blood. Okay. I will use something that will reduce, that will remove oxygen from the blood. Now the blood looks like a carbon dioxide, decreased oxygen condition. Now cells will turn to sickle cell. Is it clear? This is one approach. And second approach is very interesting. I will lyse the RBC. First I will lyse the RBC because this is the issue with the hemoglobin, not with the red blood cell. I will lyse the RBC or I will destroy the RBC with the help of a saponin. There is a reagent called saponin. Saponin is just like a bubbles reagent. No, you will blow bubbles, right? Soap, liquid oil, simply. Okay. So first I added saponin. Saponin lysed my RBCs. Now I will add a reducing reagent. That reducing reagent will convert my hemoglobin S to polymerase. They will precipitate. My HbS will be precipitated. Keep in mind, normal HbA won't get precipitated. HbA won't precipitate in reducing chemicals. Only HbS will be precipitated in reducing chemical. Okay, I will use a reducing chemical. Here the reducing chemical will be dithionite. Dithionite will remove oxygen from the blood. Dithionite is the reducing chemical. This will make crystals. This precipitate is HbS crystals. Okay, HbS crystals. HbS is the HbS crystals. They say pneumatic liquid crystals. Some technical names are there. Pneumatic liquid crystals. Some HbS crystals will be formed. Yes. These crystals, listen first. These crystals will make the liquid turbid. It will make the liquid turbid. Okay. Water will be converted to somewhat milky texture. Okay, turbid texture. Aloe vera texture. You got my point, right? Aloe vera texture. Now, turbid. In HbS, the liquid become turbid. Now, let us see a turbid sample. There are two types of samples: a turbid sample and a water sample. This is water sample and this is a turbid sample. There is a water sample and there is a turbid sample. Okay, imagine like a glass rod. Okay, now what I will do is I will keep a ruled paper. I will take a ruled paper, and if I keep my water here, can you able to see the rules? We can see rules in a water test tube. But a turbid tube, we won't see the lines because the liquid is turbid in nature. This is how the test will be done. What will be the results in HbS patient? It will be turned into turbid. That means we can't see any lines. No visible lines indicates positive for sickle cell test. That's it. This is sickle cell solubility test. And a HbF, hemoglobin A, in case of HbA, it won't be dissolved, it won't be turbid. In HbA, we will see lines. Normal patient samples, we will see lines. Whereas a sickle cell patient, we won't see the lines. Because their HbS was precipitated to make a turbidity in the test tube. This is called as sickle cell solubility test. This test is called as sickle cell solubility test. Sickle cell solubility test. Yes. Any doubts now?
Please explain.
Yes. From where I need to explain?
So start.
Yes. We understood that HBS, properties of HBS. HBS under decreased oxygen will be converted to a sickle shape. HBS under lack of oxygen converts to sickle shape. HbA, I'm parallelly drawing comparison with normal one. A normal hemoglobin, decreased oxygen, it still maintains biconcave shape. Whereas in sickle shape, first it looks like a biconcave, decreased oxygen, sickle. Now we need to mimic this test in a slide or in a experimental. What I am doing, I am taking this sample, this HBS sample, biconcave shaped cells. To this sample, I added a saponin. Saponin lysed my RBCs. RBC lysed and RBC released my HBS. HBS hemoglobin came outside. Now how I will detect HBS? I know that my HBS will be under deoxygenated condition. HBS will be precipitated. For that to remove oxygen, I used a chemical called dithionite. Okay, dithionite. Dithionite. I used dithionite. So oxygen removed. So my HBS got precipitated. This precipitated caused turbidity. How I will check the turbidity? I will check turbidity by using ruled paper. I will keep test tube over the ruled paper. If there is, if my sample has turbid, I won't see the lines. Disappearance of lines indicates positive for sickle cell test. Appearance of line, disappearance is the positive and appearance is negative to the test. That's it. Any doubts? These precipitate crystals are called as pneumatic liquid crystals. There is a scientific term for this precipitate. That precipitated crystals are called as pneumatic liquid crystals. So there will be many names for many phenomena. But the concept is this. Any more doubts?
Sir, we discussed on this second this one. Are we using the same redox solution as what we're using in the solubility test? The redox solution that we'll be using here, is it the same as what we'll be using in the solubility test?
Yes. The same. Actually, now I integrated everything. The same chemicals are used in solubility test. That's it. Now listen. From kit to kit, the chemicals will be changed. Instead of dithionite, they might take other chemical that will reduce oxygen. From kit to kit, the reagents might be changed. So, as per the kit that I opted for this material, in this kit, what they say is, as per the material kit, they say we need to take 20 microliters of reagent plus 20 microliters of blood sample. So, test is very simple. Take 20 microliters of reagent, then 20 microliters of blood sample, mix them in a test tube, wait for 5 minutes, After 5 minutes, what we need to do? We need to check the turbidity of the solution over a ruled paper. Non-visibility of lines indicates positive, visibility of lines indicates negative to the test.
Sir?
Parallelly, we will also keep a control. Control means, in this control, we have a sickle cell HBS. They will give HBS hemoglobin in the container. That is control. So I will check control with patient test. Control must give me positive results always. I hope you know what is control, right? Control will have actual sample. Yeah, the actual sickle sample will be there in control. That's it. Any more doubts? Confusion to Palak, I guess. Any confusion? Yes, Muskan, can you explain this in Hindi to everyone? That will be better. Come here, Muskan, come here. Once come here. Yeah. Yes, just try to explain to everyone in Hindi. Yes. You show your face to this. It will recognize. Come.
सर का मेडिकल गलत बोल दिया
No issues, no issues. See these ones. Yes. Continue. Everyone listen to Muskan.
ओके तो पहले हम 20 माइक्रोलीटर
Now,
रिएजेंट्स लेंगे
entire concept. You can.
ओके जो नॉर्मल एचबीए होगा वो ऑक्सीजन जब डिक्रीज होगा तो बाइकॉन्केव शेप में होगा और अगर जो एचबीएस होगा वो नॉर्मल कहते हैं जब ऑक्सीजन डिक्रीज होगा तो वो सिकल सेल में कन्वर्ट हो जाएगा फिर हम सैंपल में सैपोनिन को कंफ्यूज हो रही सर मैं
Don't worry, explain it. So we are adding a saponin. Saponin will lyse there.
लाइन आरबीसी फिर जो एचबीएस होगा उसमें हम रिएजेंट केमिकल डालेंगे डायटोनिन और फिर वो प्रेसिपिटेट कर देगा एचबीएस को और फिर उसके बाद जो टर्बिडिटी हमें मिलेगी टर्बिडिटी में कन्वर्ट हो जाएगा उसको हम रूल पेपर के साथ चेक करेंगे अगर हमें डिसअपियरेंस मिलेगी रूल पेपर में तो पॉजिटिव रिजल्ट करेगा अगर हमें रूल्स शो होंगे तो फिर वो नेगेटिव टेस्ट शो करेगा और जो क्रिस्टल्स जो प्रेसिपिटेट होंगे उसको हम न्यूमेटिक लिक्विड क्रिस्टल्स कहते हैं और जो प्रोसीजर है हम 20 माइक्रोलीटर रिएजेंट्स को मिक्स करेंगे सैंपल के साथ 20 माइक्रोलीटर ब्लड के साथ और उसको फिर हम 5 मिनट बाद उसको ऑब्जर्व करेंगे रूल पेपर शीट के साथ अगर हमें फिर से रूल्स अगर शो होंगे टर्बिडिटी होगी और रूल पेपर में लाइन्स हमें शो नहीं होंगी तो वो पॉजिटिव होगा अगर हमें लाइन शो होंगी तो वो नेगेटिव में होगा
Yes, very good. Yes, well done.